Good morning. Thank you very much for attending. I want to give a talk today just to present our company. We have a very innovative approach in cell therapy. I think my voice is loud enough, but anyways, I see. Thank you. I think we always like to make this comment that the stem cell revolution began with a fibroblast. And Dr. Yamanaka's seminal paper in 2012, he actually used a dermal fibroblast to generate the iPSCs that he used in his work that led to a Nobel Prize. Since then, fibroblasts have not been as mentioned in therapeutics, but we would like to change that. Just some comments here. Our goal and our mission is to harness the immune modulation potential and tissue regeneration potential of fibroblasts to develop therapies and cures for some chronic diseases. To date, we have 157 patents that have been issued and pending, with 61 that have been issued in the US as well as internationally. Fibroblasts, we are very confident in making the statement that fibroblasts are a more practical and a more economical alternative to MSCs for treating chronic diseases. They are very similar to MSCs, but they're much simpler to culture, isolate, and manufacture. They're also more prevalent than stem cells. They outnumber stem cells at about 5,000 to 1. They have the same capability of differentiating into multiple cell types, and publications have indicated this. They're potent anti-inflammatory response cells by secreting quite a few cytokines and chemokines necessary for controlling the immune system. And they also recruit stem cells and maintain stem cell niches across a wide range of tissues and organs. And also our indication and 2 clinical trials have shown that they're universal donor as allogenic. So they're very easy to manufacture and maintain for clinical use. Just a mention of our company leadership, about 60 years of combined experience in pharmaceuticals, medtech, and biotech. And our board of directors are seasoned executives that are currently leading medtech, biotech, and pharmaceutical companies. And our scientific advisory is world-renowned for their work in stem cells, as well as tissue regeneration, cell therapy, and of course, Kate Rubin, the Artemis mission astronaut who was the first person to sequence DNA in space. We're very proud of their achievements and being in our advisory board. Just because we don't have much time, I'm going to briefly go through some of the indications that we are targeting in our pipeline. Cybercell, that is for degenerative disc disease. We have been given a cleared IND clearance by the FDA for a phase 1/2 clinical trial, which we hope to initiate in 2025. CYMS-101 is our immune modulator product for treating multiple sclerosis. Our CYW628 is for diabetic foot ulcers. We do have some early phase for thymic involution reversal, as well as for psoriasis. I'm just going to go through some of the preclinical and clinical results for multiple sclerosis, which indicates how potent of an immune modulator these cells are. In rat models for AAE, for multiple sclerosis, the fibroblasts used as single cells as well as an organoid stimulate expansion of T regulatory cells. So we have compared these against adipose-derived and bone marrow-derived stem cells, and they are significantly more potent, both in vitro and in vivo, in expanding T regulatory cells. At the same time, they're potent inhibitors of reactive T cells, both in vivo and in vitro, in comparison to stem cells. They're also more potent inhibitors of dendritic cell maturation in multiple than MSCs, but they are also very potent downregulators of inflammatory markers such as CD40, CD80, and CD86. At the same time, they significantly upregulate anti-inflammatory response in IL-10, IL-1RA, as well as PD-L1. And this is all in comparison to bone marrow and adipose-derived stem cells. They're significantly more potent immune modulators. Looking at how it impacts multiple sclerosis, this is on an EAE rat model. We've noticed that in the cerebral cortex of EAE-treated rats, after 31 days, we noticed significant improvement in myelin expression and myelin presence. Our data has also indicated superior oligodendrocyte expansion as well as myelin expression in these rat models. This is, again, comparing it to stem cells. They're significantly more potent. And looking at microglia expansion in fibroblasts, this is in the brain of EAE-treated rats. We see a significant reduction or inhibition of microglia expansion in the rat EAE model, and we notice the same thing in the spinal cord. And looking at the severity of EAE-treated EAE rat models treated with the spheroid fibroblasts, as you can see, significant improvement within 7 days of the EAE score, practically reversal of the EAE starting at about 7 days. As you notice with adipose-derived stem cells, you see some relapses. We saw no relapses with these mouse models, with these rat models at all. So just as a summary, we see significant increase in T cell expansion, T regulatory cell expansion, and suppression of reactive T cells. We see inhibition of dendritic cells and downregulation of cell surface markers responsible for inflammation, and upregulation of anti-inflammatory cytokines. We see significant suppression of microglia activity, as well as significant increase in myelin expression in both the spinal cord and the brains. Our 5-patient phase 0 clinical trial indicated no adverse events during the 16-week treatment with intravenously injected fibroblast cells. We did see some efficacy as a secondary outcome, but the number of patients was too small for it to be significant. But one thing to note is that during that 16-week monitoring period, the patients that were treated with fibroblasts exhibited no further deterioration or no episodes. It's 16 weeks, but our hope is that our next clinical trial, phase 1/2, we will target a much longer, of course, for about a year and a half follow-up. Our next target is the diabetic foot ulcer. These are chronic wounds. And as you're probably aware, fibroblasts play a role in every single stage of the wound healing process. So it would make sense to see if fibroblasts could help improve or heal chronic wounds as well as other type of wounds. So we used a diabetic mouse model. We introduced wounds on the back of these mice and rendered these wounds chronic by using a catalase and glutathione peroxidase. And after the wounds became chronic, we introduced fibroblast spheroids on the wounds and monitored the wounds until they healed. Just giving you an indication of what these wounds look like, we compared it against the FDA-approved product for DFU, which is that competitor G at the bottom. And as you can see, within 4 days of treatment with spheroids from fibroblasts, we saw a 60% reduction in the wound size, wound area. Whereas we only saw about a 30% reduction with the FDA-approved product. And looking at the percentage reduction in size over time, monitored for about 14 days, we saw an 87% reduction with the fibroblast spheroids within that 14-day period, and we only saw about a 60-something% with the other product. So this is only a single administration of a fibroblast. We are investigating in the next phase multiple administration to see how quickly it could heal the wounds. And also, even with fresh wounds on diabetic mice, we see a significant improvement in wound healing. We see the wound closure and scab release and reepithelialization within 5 days with fibroblasts, whereas with the other product, we're seeing it at day 8, and of course, no treatment. It usually takes about 14 days for epithelialization to occur. And here's an image of the spheroids on the surface of a wound. On the left panel, you see the spheroids just added to the surface of the wound, and within 6 hours, you see the spheroids migrating outwards. This is not grafting. This is releasing of all the growth factors, all the cytokines and chemokines necessary to initiate the wound healing process. So our next target is psoriasis. This makes sense because psoriasis is another autoimmune disorder. It's mediated by the cells of both the innate and adaptive immune system. So since we had such great response in our MS in terms of immune modulation, we wanted to see if this would be impacted as well for psoriasis, another autoimmune disorder. So we looked at an animal model, a mouse model for psoriasis. This uses amoxicillin, which is treated daily on the back of a mice, and this gives the symptoms that are very similar to human psoriasis. And we continue this administration, and once we see the scoring of the psoriasis reach a moderate level, we then applied intravenously injected the fibroblast to see how much of an improvement it makes. And as you can see, after day 4, after introduction of the fibroblast, we're seeing a significant improvement of the symptoms of psoriasis in these mouse models. And as just looking at it, the whole blood count, we're seeing an improved blood count within that period of time as well in these mouse models. And we're also seeing reduced severity of psoriasis within 35% in a single administration. Our next phase of this study, we're looking at multiple administrations to see how quickly we can reduce the symptoms even more quickly. So on a side note, psoriasis is not involved in chronic inflammation, which leads to an enlarged spleen. One of the things that we noted in these mouse models is that because a mucor mold initiates a chronic inflammation response, increases the size and volume of the spleen, one of the things that we noted was a reduction in volume and size of the spleen, which was highly significant. And we're considering, and we're also testing the methodology for alleviating some of the symptoms of chronic inflammation, which is the cause of so many of other clinical disorders. We're also targeting thymic involution. As many of you would probably know, thymus is the teaching center of the immune system, but in humans, it has a 15.7-year half-life. That means by the time you're in your 60s, you've lost about greater than 90% of the function of your thymus gland, which means as you get older, you become, you reduce much, your response to vaccination becomes reduced. You have an increased chance of cancer and an increased chance of autoimmune disorders. We are currently, we've started an animal model to look and see if we could use either thymic fibroblasts or even human dermal fibroblasts to reinitiate the thymus gland and rebuild it. Hopefully this will give a much better life expectancy or life quality of life for some of the patients, elderly, which is great. So in summary, we consider fibroblasts as a novel platform technology for developing treatments and cures for chronic disorders. Very similar to stem cells, but a lot easier to obtain, more ethical to obtain, more easy to culture, more easier to maintain. And they've been clinically proven safe as an allogenic treatment in both degenerative disc disease in our trial, as well as for multiple sclerosis. And they have a significant potential in treating quite a few chronic disorders, including autoimmune disorders. And we maintain a very healthy and extensive patent portfolio of the use of fibroblasts in treating multiple chronic disorders. So thank you very much.